All right, grade 10s, here we go. We're starting a new unit, basically a bio unit, and we're going to start off with chapter 7. Some of this is going to sound very familiar from your middle school years. Chapter 7 is called The Basis of Life. It's going to spend a lot of time talking and looking at cells. So, of course, this title slide here has hopefully you recognize this cell here. This, of course, is your red blood cell. It is the most numerous of all the cells in your body. You have trillions of them. And of course, their purpose is to carry oxygen with their hemoglobin. And they are really one of the only cells, if not the only cell in our bodies that does not have a nucleus. All right, so, and if you're carrying on in biology in grade 11, we will do a whole chapter or two about blood and the importance here of these red blood cells. All right, well, first thing we're going to talk about, because we're kind of first going to look at what we call our cell theory. What is it that we sort of know or understand about the basic unit of a living thing, which is the cell? So we have to cover what's called abiogenesis, because in the olden days, it was once thought, let me adjust this so you can make sure we've got the whole screen here. In the olden days, this was very much the prevailing thought, abiogenesis, that you could actually get life from non-living matter. All right, so of course that had to kind of happen in some way, shape, or form, either supernaturally, as I would subscribe to it, because you had non-living, and then all of a sudden there was life. It was first proposed by Aristotle, and this would come from observation. Maggots would appear on decaying meat, or you would lift up cloth from the corner of your kitchen and then a mouse would run out. And or all of a sudden, as we had a lot of rain lately, you'd see all these little worms wriggling around, and it's as if it rained and then poof, there's all these worms and living things around. So that's called abiogenesis. It's also known as spontaneous generation. But none of these theories were really tested scientifically, because scientific testing was not in vogue in ancient times. It really was a part of the Enlightenment, when turn around 1500s, when people actually start to test scientific theories. So let's talk about a few players, a few scientists that were involved in either mainly discounting this idea of abiogenesis and being proponents for biogenesis, that life comes from life. So here we have Francesco Redi, and he does a famous experiment where he has meat, and he has different containers, and some of the containers are covered and some of the containers are not covered. And what he, of course, sees over time is that in some of the containers, there's maggots on the meat, and in some of the containers there are not. Of course, it's the covered jars. So I think I have a picture coming up here. Well, it's really hard for you to see on this video. But obviously we understand that the maggots are the larvae of flies, and that on the open containers, the adult flies were able to come and lay their microscopic eggs in the meat or on the meat, and that over time they've developed, the eggs have hatched, and now we have larvae, and that we can see them. So there was no spontaneous generation. This was an experiment that proved biogenesis, that those maggots did not just suddenly appear from non-living matter. They came from adults, from life. Then we have Louis Pasteur, who is famous for the process of pasteurization, which you may have heard of before. It's used when making some beverages, or especially milk. It's the idea of heating up something to kill all the bacteria so that it's more safe to drink and safe to store. So we can thank Louis Pasteur for this, right? And he also did an experiment. We call it the experiment with his swan-necked flask. So he basically had a broth, and he put it in a flask, and it's called a swan nest flask because of the shape of the tubing there. And then he was able to, he heated up the broth to sterilize it. And then, of course, the steam and the air were forced out of an open end. And then we have sort of a trap here, a bend like you kind of have under your sinks or under your, what a toilet has, so that it actually kind of protects from the open air. And so any dust or microorganisms would trapped here. And for a very, very, very long time, for as long as you wanted, the liquid would stay sterile and you wouldn't get any living things in that liquid. However, if you tip the flask and you allow this liquid to come in contact with the air or with the trap, it would not take very long at all before you had living things. So again, we only have living things once that broth came in contact with bacteria or the air, which would have the living particles or living things traveling in it. And they were microscopic, of course. All right. Now, other scientists involved in coming up with our cell theory. So an English scientist, Robert Hooke, in 1665, he discovers cells while looking at a very thin slice of cork. He describes the cells as tiny boxes or a honeycomb. He actually coins the term the cell, not because of prison cell exactly, but monks used to live in what they called cells in a monastery. And it was kind of like little individual square rooms. All right, so, and now he thought that cells only existed in plants and fungi, that animals, it would be kind of a different deal. Anton van Leeuwenhoek comes along in 1673. He's kind of famous for making a weird looking paddle-shaped microscope. And he observes pond scum. He also looks at dental scrapings off of teeth, blood cells from fish and other places. And so, and now he called what he was observing animalcules. Now, guys, these are kind of key vocabulary terms. So when you get these matching questions, questions on tests that kind of say who said what, you key in on these very kind of specific terms. Hooke, first to observe the cell. Van Leeuwenhoek, first to use the term animalcules and see small cells in animal matter. All right, so he helps kind of pave the way for cells being the basic functional unit of all living things, plants and animals. So, with the help of a couple of German scientists that you'll see a video about when you check the video clips, there's a nice little video there that goes through all the scientists involved in coming up with the cell theory. Schleiden and Schwann were German scientists. One kind of looked at plants, one kind of was looking at animals. And kind of together they sort of realized, hey, you know what? Plants and animals have the cell as their basic functional unit of life. We need to talk about Virchow. Virchow is a Russian scientist who notice in 1858, so notice how the cell theory is taking almost a couple of hundred years to develop. He saw that cells would divide under the microscope. So that added the third component of the cell theory, which is that life comes from life, or cells come from pre-existing cells. Okay, so there you go. These are the three basic parts of the cell theory. All organisms are composed of one or more cells. The cell is the basic unit of life in all living things, and all cells come from pre-existing cells. Okay. Now, let's turn our attention to microscopes very quickly. Guys, when we talk about microscopes, you don't have to memorize everything there is to know about each microscope, even though we have fairly well-labeled diagrams here. You really just have to be able to compare them in terms of their magnification and in terms of their clarity or resolution, and then that's really all you would need to be able to do. And again, you're kind of going to get a matching set for your quiz or for your test. So let's start with a basic light microscope. These are the microscopes that you're most familiar with in most science classrooms. They work on the visible light, so they're only really good for things that light can reflect off of. Maximum magnification really of about 1,500 times. They use a combination of lenses for magnification. So just a reminder about the electromagnetic spectrum. Light microscopes are using waves of light. Remember that the only reason you're able to see something is that light strikes that object and then comes back to your eyeball. And our eyes are designed to just capture this wavelength of radiation, visible light. But there's lots of different forms of radiation. And that's why we can make different microscopes. Different microscopes can pick up on different wavelengths of energy. Really, the smaller the wavelength, the more magnification and often resolution we can get when using microscopes. Now, only really visible or light microscopes allow us to see colors of things. As soon as we get outside of this range, we can magnify and get really good resolution and detail, but we would lose color. All right, so most of you are going to be familiar with electron microscopes. So an electron microscope gives us much greater magnification, gives us much greater detail, but really we wouldn't get color because we are now outside. We are using energy with a wavelength that is shorter than light. So if you do see a colored version of an electron micrograph, a computer has added the color to it. Okay, kind of like taking an old black and white and adding color to that, kind of artificially. So there's two kinds of electron microscopes. We have a transmission electron microscope, which kind of just gives us a 2D image, and the electrons would actually kind of go through, whereas a scanning electron microscope is going to reflect, and it provides a 3D image. You can see we get much greater magnification with the scanning electron microscope than we do from the transmission, and you would need to use a very, very thin sample of your subject matter to use an electron microscope. Now, there is a CLSM, a confocal laser scanning microscope. Now, this is kind of going to work a little like a CT scan or a CAT scan. You can use a thicker specimen. It's going to take 2D pictures, so it might actually be able to rotate around and take slices, kind of images, and then again, kind of like an MRI or a CAT scan, a computer can put those images together to give a really good 3D image of the object. So that's using laser, which again, high intensity light. So it's really a form of the light spectrum. And now this you just have to know as our most powerful microscope, called the scanning tunneling microscope. It can actually, so again, it's using electrons, and the electrons are bouncing off, kind of like sound waves would bounce off objects from, like, say, the ocean floor. If you were mapping the ocean floor, you can use sound waves. So it's similar to that, except it's using electrons to bounce off of a surface. And then again, we might even, or as you can see here, actually see the shapes of molecules. So we're getting down to a molecular level. So that's a very high magnification. Now, in this section, which I think is 7.2, there's some other things we discuss in relation to cells. We know that DNA directs all the activities in a cell. It's in the nucleus of a cell. And a segment of DNA is called a gene, which is just really instruction, units of instruction that the cells can follow to do whatever that cell needs to do. The gene sequencing has taken place so that we can figure out the group of bases. Now, guys, you might remember in grade 9, you would have studied a little bit about DNA. DNA are strands of what we call nucleotides, and every nucleotide, think of it like pearls on a pearl necklace. And so they are repeating units, and they only differ by what's called the base component of the nucleotide, and all these bases have letters, and you might remember them. They're A, C, T, and G, and that forms a 4-letter code, and it's the sequence of those letters that form a unit of instruction, which we call a gene. And so again, here's sort of our picture of the double-stranded DNA molecule, which is held together by those base pairings. You might remember that. A pairs with T and C pairs with G. That sequence will code quite often for a protein, something the cell has to make. All right, so just a little review there. Now, cells, of course, divide. So if cells divide uncontrollably, again, something has gone haywire with the instructions for cell division, it's part of that genetic code we were just talking about, then cancer may result, or at least a tumor, but not all tumors are cancerous. If it's cancerous, then not only does the division happen uncontrolled, but those cells are non-functional. They've divided so fast that they're not going to do what they're supposed to do. They may even have the ability to not adhere to one another. Then they start to break away from the main tumor and they migrate. That's called metastasis. That's why someone with cancer in one part of their body may end up with cancer in many other areas of their body. So why you always want to catch it early if you can. And again, I told you there's a genetic factor to cancer. The genes we think that sort of regulate the possibility of cancer forming are called oncogenes. So that prefix onco, it's often used, for example, an oncologist, a cancer doctor. All right, so there we go. Now viruses, like we want to talk about viruses. So right now, of course, being in the midst of this worldwide pandemic, everybody's talking about viruses. So what's a virus exactly? And boy, because it's kind of out there in society, you may very well know. So let's talk about viruses. So are viruses made of cells? What's a virus? So really, guys, a virus we could almost consider not to be a living thing. It's not made of cellular material, like it's not actually a cell. It needs cells to survive, and it really has one job, and that's to make copies of itself. It hijacks other cells that it's able to penetrate and it basically inserts instructions which say, make more of me. And so there you go. And so this virus that we're dealing with now, this COVID, which causes the COVID disease, the virus actually has its own name. You may see it every once in a while. It's a SARS-related virus, so it's like SARS-CoV 19, and so it's just a new coronavirus. And so there's some good videos that will explain all about it. So really, it's non-cellular. It's simply made up of some genes, often RNA strand, which is a strand of different nucleotides than DNA, and it's just surrounded by a protein coat. And it can infect cells. It deposits the gene into the nucleus of a host cell. The cell makes more virus, and there you go. And then you could have a worldwide problem on your hands, depending on how contagious it is and the symptoms, etc. And guys, the reason we're having such a problem with this virus is it's highly contagious, and unfortunately, or fortunately, it doesn't make everybody super sick. So really, you can be asymptomatic. So you're carrying the virus, you're shedding the virus, and you have no idea. Whereas if the virus made you really, really sick, like SARS, many years ago we had SARS here in Canada in the Toronto area, people would get very sick very quickly, and so there wasn't that opportunity to really spread. And of course, when you're shedding the virus, there's different, sometimes you can't make anybody sick until you're really sick yourself. But with this virus we have right now, you can be asymptomatic, and so you can be what they call a super spreader, because you go around and you have no idea, and you're not that sick yourself. So highly contagious, and almost doesn't make certain people sick enough to slow it down. So one of the reasons we have a problem on our hands, literally, sometimes on your hands. So wash your hands. Okay. All right. Now prions, guys. So prions are proteins that can become harmful. So they're kind of out in the environment. Now, mad cow disease, being in Alberta here, mad cow disease, big problem. Sometimes we get these flare-ups. That's thought to be caused by a prion. So it's just a protein. Again, it's not a cell. It's not a living thing, really. But again, it gets into the animal system, and it causes problems. Different prions cause different problems, and in the case of mad cow, it's in the brain of the cow, and it stumbles around and doesn't behave like it's supposed to. If we want to study cells, we can make what's called a cell culture. So you can use a petri dish and you give it the nutrients it needs, maybe an agar solution, and the cell. So you can take a small sample, and then if you want to study it and study lines of the cell, then you need to grow it in what we call a culture. So that's just vocabulary, all right? And then, of course, you may have heard about stem cells. There's going to be a very good video clip in the classroom, Google Classroom, that will talk about what stem cells are. essentially they're blank cells. They're cells that have the ability to become any other kind of cell in the body. And of course, there's a range where we use terms like pluripotent or totipotent. Again, if you look at the video, it'll clear that up. I'm not going to be asking any questions that are that detailed, but just know that a stem cell, so for example, of course, we have embryonic stem cells. When you were one cell and then a small ball of cells, all of those cells are what we call embryonic stem cells because they have to specialize eventually and become the different organs and then work as a system at some point. And so there's lots of science going on about trying to collect stem cells, study stem cells, reproduce stem cells, and if they're your very own stem cells, the advantage is you won't reject them. So we could use your very own stem cells to treat your own diseases later on in your life. And that would be the advantage of being able to use stem cells. You have stem cells even as a young adult. Me as an adult. Our skin cells, our blood cells are all come from one cell in our bone marrow, which is a stem cell, because there's many different kinds of blood cells, but only one cell in our bone marrow differentiates into those many different kinds of blood cells. I think that's it, because the rest of this chapter, guys, is going to deal with plant and animal cell structures and functions. So you can look forward to that at another time.